You found the skeleton. How would you tell it's a dinosaur? You personally, how would you decide?
Well, interesting question. I don't know. I'd have to do some research on that.
Hey there, YouTube, the Dapper Dinosaur here. We're going to learn about Darwin, I guess, which is a bit weird since he's just a scientist from a long time ago who really only matters as the guy who came up with a couple mechanisms for evolution and isn't exactly on the cutting edge of science. But, oh well, here we go.
Origin of species. He thought that was the key problem, to describe where species come from.
I don't know about the key problem, but it certainly was a problem he was interested in, and hence he gathered data and made a hypothesis about it. Turns out he was basically right, although in some details he was wrong, and there's a lot more to it than he knew about.
And there's a certain way of describing how the changes give rise to new species. It's a fourth meaning of evolution. None of these is what we're talking about.
Really? You could have fooled me, because between common ancestry and the mechanisms of evolution, I'm not sure what else we really could be talking about.
None of these is what we are talking about. One second. The fifth meaning, when we are talking about, is this. What is the cause of the changes? What makes the changes happen? What's the explanation of the changes?
Mutation, selection, and drift, all of which are already observed in labs, in the wild, and in simulations.
The theory of evolution says the major, the most important, the primary cause of the changes is random variations, which then are subject to selection. The random variations have effect on how the changed organism survives. And these have to be heritable. That is to say, they have to be inherited from organism to organism. And that's what does it. Yeah, there's some debate about other contributing factors, but the major factor that does the job is random variations plus selection.
Actually, I would argue that in the long run, drift is more important than selection in terms of the actual genomic content of populations, but selection is also very important for allowing such populations to persist.
That's what we're talking about. When we talk about probability, it's that description of evolution that we're talking about.
But probably only applies to the mutation side of that because selection is decidedly non-random. We can't just ignore the non-random processes. We're back to my reaction to fusion simulation. The initial setup where the two chemicals, or in my case numbers, are random, but what happens to them over the course of the simulation is not. Given the same seed of numbers and the same grid to propagate across, you'll get the same simulation each time.
You see the envelope of life. You say that there were changes and gradual changes. All that you could conceivably verify from the fossil record.
Which we have.
Without an explanation, without a cause, without an engine, without knowing what makes it happen that way. That's a separate discipline, a separate study.
Yes, in fact, this idea is faunal succession, and it was well known before Darwin ever published his book. It was often explained by progressive creation before there was a well-understood mechanism for evolution, but coupled with the fact that we can watch in real time mutation and selection occur in a lab, like in the long-term Lenski experiment, and in the wild, like in Darwin's finches, that means it's a solid explanation for the evidence of the same change in the past.
What we're talking about is the engine that makes it happen. Now, the reason that's important is because the engine that makes it happen is founded on random variation. Selection only preserves the ones that are successful and eliminates the ones that are not successful. You can't create anything with selection. You can only cut out the unsuccessful ones with selection.
Right, this is no surprise. It is well acknowledged by everyone in the field of evolutionary biology. Selection does not produce novel genetic sequences or novel traits. This is done by mutation. Selection simply then acts to eliminate or preserve and spread around these sequences and traits.
Behind selection is something that's generating new types, new features. That's where it's an unguided, unmanaged, unfocused. I know the word random is tricky. The word random has all sorts of technical meanings. No one has a good meaning for the word random. Too bad, it's another problem. But it's accidental, unguided, unfocused, no intelligence behind it. Then I want a probability. That's no different from the gun spinning on a pivot or an object landing on the X 3 times. Is something which is supposed to happen by accident. Well, I want a probability for that.
For what? The existence of mutations? It's 100%. For the probability of any given mutation? Well, that's a function of the kind of mutation and its locus, but it is in principle calculable. For the chance that a particular population would produce any mutation that resulted in a positive or negative selection, well, we'd need to know how much of the sequence space could result in such selection, which means that our estimates would always be low, but that's also calculable. But I still don't know what specifically he wants a probability for. It's like me asking him for what the probability of Judaism is. I mean, it's 100% probable that Jews exist, because I can go look at some of them, talk to them, etc. What are the probabilities that Judaism would develop as it did after the destruction of the Second Temple? I mean, probably low, but not zero. The probability that the stories contained in the Tanakh are historically true? Well, that's going to vary from essentially zero for some to nearly 100% for others. See what I mean? You can't just ask for the probability of some big thing and then get back a meaningful answer. He needs to be much more specific. The chances for a given mutation occurring are very low. The chances for mutations occurring at all is 100% if the population is still reproducing. The chance for selectable mutations is experimentally shown to be very high because it's trivial to induce natural selection in laboratory populations and then show that selection has in fact altered the frequency of certain traits in that population.
And if you don't give me a probability, there isn't enough reason to accept that it's true. Period.
If you don't answer my malformed question, I'm going to reject science is certainly a take. I don't think it's a good one, but it is one of the takes of all time.
Questions up to here. Now, I've challenged people with this. I've told them, write to your biology professors, write to your philosophy professors, get me an answer to this. I know there isn't an answer because Thomas Nagel put it in a book which caused howls and shrieks of protest, but nobody answered it.
When someone asks a question that is not even comprehensible, it's not surprising that no one answers that question. For example, if I ask, how is yellow, there isn't much of an answer to be given. If I ask, what is the probability of electrons, that similarly has no answer. The probability that evolution occurs is simply 1. The probability of things within that is going to vary.
Nobody even tried to answer. Nobody said, oh, you say there's no probability? Here it is, A, B, and C. Nobody even tried to do that. How's the protest at all? But nobody answered that point.
Well, if it's the same point that David is trying to make, then I expect the howls or protests were basically the same as mine. You can't answer a question that doesn't make any sense. If you want to know what the volume of Earth's gravity is, there isn't a meaningful answer to be given. If you want to know how many pints make up a mile, that similarly has no meaningful answer.
And as I say, it's not a new point. It's been going on for decades. I think that's conclusive.
The only thing I can conclude from this is that this guy has absolutely no idea what evolution is, how statistics works, or how science works. Which wouldn't normally be a problem, except that he made a video in which he attempts to spread anti-science belief, and that's not okay.
Try it out. I'm sure you know true believers. I'm sure you know people who, willing to live and die for evolution. Ask them that question.
Please don't. Please ask a question that makes sense.
I've been talking about this like this way for 20 years. No one has even offered an answer to the question.
He could have been talking about this way for 200 years. If you ask a question that doesn't mean anything, you're not going to get an answer. Let me ask you, how long is a kilogram? When are letters? Where is acceleration?
I think that's very important. Are we together so far?
Nope. I'm lost.
Okay. Now, there's something else called vestigial organs.
That's a Matt Powell level topic change, but okay.
Vestigial organs are features of the body that either don't do any job at all, or they do the jobs badly. Vestigial usually refers to things that don't do any job. Or they're badly designed.
No, that is a bad definition. It might work fine for babies, but adults who want to actually meaningfully discuss evolution need to do better. Vestigial organs are those that no longer function effectively for the primary use which homologous organs in ancestral and related populations have the organ. So, for example, an ostrich wing is vestigial because in other birds, including the ancestors of the ostrich, the wings are primarily organs of flight, but ostriches don't use them for that, being flightless. That doesn't mean that they're useless. They help the animal communicate and keep balance.
And here there are two failures, a philosophical failure and a factual failure. Let's see how it's supposed to go. Let's say there are features of the body that serve no purpose, which would be an indication of bad design. A good designer doesn't put in things that don't have any purpose. Or features that are badly designed, that don't do their job in the best way that they could. We could easily think of designs that would do it better.
Neither of these things would indicate vestigiality on their own. For example, cats with polydactyly generally get no real use out of their supernumerary claws, but those claws aren't vestigial because there was no polydactyl cat ancestor with use for those claws.
Let's say that's true. So what? What's that got to do with evolution? What's it got to do with evolution?
See, the thing is, I can't tell if he's asking what do useless organs have to do with evolution, or what do vestigial organs have to do with evolution. I guess I'll try to answer both. Useless organs generally do not persist in a population because they cost energy to make, can become injured, increase mortality, and there is no selection pressure to keep them. As a result, if we could see the persistence of completely useless organs in many organisms that are not vestigial, that would be evidence against evolution. We generally don't see this, at least in the wild. Domesticated animals, like those cats with too many fingers, are generally shielded from some of the harms that they might otherwise suffer from various genetic problems. What vestigial organs have to do with evolution is that they are explained and predicted by evolution, but other options such as special creation do not predict them, and can really only accommodate them by having a designer that wants to design things to make it look like evolution occurred, since in known instances of intelligent design such things generally would not be included. Like, if you're designing a car for northern Greenland, you probably don't put in an air conditioner, but seal the vents to it or something so it can't function as an air conditioner. Even if the added weight gives you better traction or something. Now you can argue that Hashem, since this guy is Jewish, just doesn't design like humans do. Okay, but if we're going to accept that he can design deceptively and poorly, it becomes essentially impossible for the evidence to indicate design in the first place, simply on the basis of the actual traits of organisms. Instead, to indicate design, you would have to disconfirm every single non-design possibility, even the ones no one has thought of. Good luck with that.
So the proposal here is double. Number one, it supports evolution, and number two, it rules out an explanation in terms of a creator.
I wouldn't actually say that it rules out the possibility of a creator, just that it indicates against design by any non-deceptive creator.
It rules out the explanation in terms of a creator because creator, we assume, could do better than that. Creator could make excellent design or perfect design. And if it's clear that the design is bad, then it's not going to be a creator who did it.
Well, not one with the qualities normally ascribed to the creator in Abrahamic religions like Judaism or Islam. Although others like Mandaism might more easily accommodate a creator who just kind of sucks at creating and also wants to trick everyone, because in that mythology the world wasn't actually created by the object of worship of Mandaism, but rather by the combination of an angelic figure, Tahil, and the queen of the underworld, the evil Ruha. Then again, Jews don't have to believe in any gods, although it's hard to see why an atheist Jew would want to argue in favor of a creator over and against evolution. That seems like the purview of the theistic types. Of course, just from looking at David Gottlieb, I feel like he'd be the one to say that atheist Jews don't count as Jews, but hopefully I'm wrong.
Whereas on the other hand, evolution can explain it. Because how is evolution supposed to work? You got a thing that's waddling around, trying to get food and reproduce and run away from its predators and adapt and...
Way to poison the well by describing the very existence of life forms as silly. Maybe that's not the best metaphor for this video. Well, I only mean it metaphorically, and I don't know what other metaphor to use. Sorry about that. Fun fact, most of the times that Jews in medieval Europe were accused of poisoning wells, it was in fact because of diseases that weren't even waterborne in the first place. So that's a thing. And even in cases when it was a waterborne disease, it's not like the people who hadn't figured out sewers needed outside help to spread that kind of disease. It's always been easy to blame those you see as others when an otherwise inexplicable evil befalls you. But that doesn't make it right.
And there are other things running around, competing for food, and there are things that want to eat it, and things it wants to eat, and it's just, doing its best in its circumstances. If it succeeds, that means it succeeds on the whole.
I don't know what that means.
The fact that it succeeds doesn't mean that it's perfectly designed. It just means that on the whole, with its good design features and bad design features, on the whole, it's good enough to keep going.
Yeah, that's true. Natural selection selects those organisms that are good enough. Rarely do individuals actually reach the peak of fitness possible in their environment. Even when they do, it means that simply because any variation away from their traits will of necessity be less fit, and because their offspring are guaranteed to at least have some traits that are different, that the organism's descendants will on the whole move away from that fitness peak anyway.
Now suppose you have a mutation and that changes it a little bit. Makes it a little bit better. So then it'll be better. And then it'll be better than the old ones, and outcompete the old ones and do better in the environment. No one thought that it's perfect.
Correct. That an organism is better than its peers in terms of evolutionary fitness doesn't indicate that it has perfect fitness, by which I assume we mean the highest fitness possible in the current environment.
Furthermore, when you make a little step to get better, that little step to get better has to be good for the thing you're working with. And the thing you're working with might not tolerate steps to get better that don't help it, because it's carrying along all the stuff that it has so far.
I'm not really sure what's being said here, which seems to be a theme with this guy. He just says words that don't really seem to correlate to anything in evolutionary biology. If a new trait is beneficial, then it's beneficial. What other stuff can't tolerate this trait? Other traits? Then the new trait isn't beneficial. This is the problem with trying to discuss evolution or other technical fields with people who don't have the first clue.
Eyes. Are eyes useful?
In many environments, yes. In others, like a deep cave, often not.
Sure, but not under your armpit.
Yeah, probably why most organisms have eyes on their head, or at least the end of tentacles, and not under armpits. But I'm not sure what the point is.
That's not going to be very useful. Because if you have to see things, you're going to walk around like this. The saber-toothed tiger is going to get you for sure. Right? Not everything helps. A thing helps because it grafts onto where you are. You aren't necessarily carrying along with you everything you've got. When you make a change, it's in addition to everything you've got.
Yeah, it's well understood that evolution is constrained by the current morphology of organisms. Bats aren't going to suddenly grow spinnerets, even if it might be handy for them to have spider webs of their own. All evolutionary change is a modification of what came before, and on the ground, in real time, it always looks fairly minor. It takes deep time to make things look like real novelties, but even then, if you could watch it all happen, you realize that, like the author of Ecclesiastes says, there is nothing new under the sun.
Now, let's imagine, here's an example that I actually use. There are animals that are underground which have eyes but are blind. Because the suggestion is they're living underground. Living underground where there's no light. Where there's no light, there's no use for sight.
Beyond that, eyes are easily injured, and so are a risk to any animal with them. But if that risk is outweighed by the usefulness of being able to see, they'll persist. But for, say, a mole, since there is a risk to the eyes and basically no benefit, they tend to go away through evolution. And during intermediate periods, they will be small and degenerate, perhaps even completely covered by skin so they're not exposed to injury.
So keeping all the neural machinery that underlies sight is pointless. You can put it to better use with smell or with digging or something else. The eyes are carried along. They had eyes before, so the evolution hasn't devised a way. Carrying the eyes isn't too expensive if the brain is disconnected from that. It's just a piece of fold of flesh, that's all.
I thought he was supposed to be arguing against evolution, not explaining why vestigial organs with little to no use may persist. I guess I should thank him for helping me do my job.
Okay, deep sea fish, same way. So you carry along the eyes. Vestigial organs are things that once were useful, but then you change your style of life. They're no longer useful because your style of life doesn't need them anymore. And evolution just hasn't gotten around to cleaning them up and eliminating them. That's what evolution predicts. When you find vestigial organs, that's evidence for evolution, and it's also evidence against the main competing hypothesis, which is that God did it.
Well, it's certainly not evidence against theistic evolution, which involves a god, but since no one predicted the existence of vestigial organs on the basis of intelligent design that I can find, it's certainly not bolstering that idea. And given that God is generally thought of as super smart and benevolent, I argue that special creation should predict that we would not find vestigial organs. But of course, once you have an omnipotent actor, you can accommodate any data, which means that the idea is unfalsifiable, which also means it has no place in science.
Both ends of this story are wrong. First of all, enormous progress has been made in the last 20 years in discovering uses for vestigial organs.
Okay, but again, vestigial doesn't require that an organ not have any use, just that it not have the typical use for such an organ.
And the fact that what used to be thought were poorly designed poorly designed organs or features of the body are really not poorly designed at all, but optimally designed.
Tell just about any human over the age of 35 that their spine was optimally designed for bipedalism, and then let them glare at you in back pain.
The appendix plays a role in the immune system. So do the adenoids.
Yep, but that doesn't make them vestigial or not. The question is, what do they do in other organisms who inherited them from the same ancestor as the one that gave the population with a potentially vestigial organ theirs? Is it what they do in the organism in question?
The eye, even Dawkins wrote that the eye proves the designer is incompetent, because you have the retina, and then you have the optic nerve that goes up to the brain, and the retina comes from inside, the optic nerve comes from inside the retina. By inside, I mean closer to the surface of the face, and goes through the retina, through a hole, and then goes up to the brain. And that means that there's a little hole in the center of the retina, and at that point you can't see, because you haven't got any retinal cells to be sensitive to the light. That's what's called a blind spot. You have a blind spot, one in each eye, where the optic nerve goes back to the retina. So, until very recently, the attitude was, what a kludge! Anybody would understand that you got to put the optic nerve behind the retina, not in front of the retina and go back through. It's ridiculous. And there are creatures that have it that way. The eye is very, very popular in evolution. According to Ernst Mayr, the eye evolved independently at least 40 times.
I don't know about the number 40, but it certainly evolved independently many times. But I'm waiting for why the optic nerve going through the retina is a good idea.
Independently. It's a very popular thing. Convergent evolution like that is something that one has to talk about, but I don't have time for it tonight. At any rate, it turns out, contrary to what they used to think, about 5 years ago, some studies were done. Having the optic nerve outside and going through a hole in the center is optimal.
Hard to see why, but let's see what he's got for us. Also, I'd love it if he actually cited this study, but alas, we just have to take his word for it, which, I won't do.
It's not a detracting factor, it's optimal. Because there are certain cells that are necessary to keep stability of the retina, and they are connected to the optic nerve, and all of them are supported by the optic nerve going back through from the outside, and that makes it optimal.
No, it doesn't, because as has already been stated, there are other options for the eye that also work, and which don't have this lack of needed stability. The fact that the tetrapod eye is stabilized by the optic nerve that comes from in front of the retina doesn't mean that that's how eyes have to be stabilized or are best stabilized. Squid have eyes that are stable, and yet their optic nerves come from behind the retina.
Furthermore, the blind spot. When's the last time you noticed your blind spot?
Almost never, because while it's suboptimal, it's good enough. You know, that thing we've already agreed is what evolution is looking for, if I can be forgiven for anthropomorphizing evolution.
When's the last time you missed the bus because your blind spot got in the way and you couldn't see the number?
Never, because buses are designed to be noticed by humans, and so they tend not to fit right into the blind spot visually. But I'm sure I've failed to notice a bug or two because of my blind spot. Perhaps even a mosquito I'd rather have crushed than let drink my vital fluids.
You're not even conscious of the blind spot. Till you're told about it, you never notice it.
That's still irrelevant to whether it's an ideal design or a poor design.
No one ever got eaten by a saber-toothed tiger because the tiger disappeared in the blind spot and you couldn't see it, and therefore it's tiny. It's tiny. And the brain fills in the background to cover the spot so you're not conscious of it.
Work the brain wouldn't need to do if the blind spot were simply not there, which it doesn't need to be.
So the whole story was wrong.
Not based on what we just heard, but also, why is this guy so upset about this? It's just people differing on the origin of biodiversity. It's really not something to get heated about.
I want to give you a little bit more detailed example of the same story where it's wrong, and this is just it's beyond belief.
I guess I won't believe it then.
What does DNA do?
Depends on the bit of DNA. Some codes for protein, some regulates gene expression, some is just a mechanical barrier to damage of other parts of the genome. Some of it spaces out genes so that they aren't all transcribed at once. Some is introgenic and can change the final product of the gene by splitting it up into smaller bits. Some of it does basically nothing useful, which is actually most of it.
Until about 15 years ago, the consensus was DNA codes for the production of proteins.
That consensus hasn't changed. It does. It's just that not all of it does.
The DNA sits there. RNA sidles up and attaches itself to a section of the DNA?
Nice use of sidle, but not quite. The RNA is built up to correspond to the DNA being transcribed. The RNA isn't just floating around, the constituents of it are, and they arrange themselves into an RNA strand as transcription goes. See what I mean about this guy not actually knowing what he's talking about?
Copies its structure, then it detaches, and it goes over to a ribosome, and there it creates a protein.
Other than the mistake I already corrected, yes, that is how RNA works. In absurdly simplified terms that ignore things like the difference between transfer RNA and messenger RNA.
The DNA's instructions for creating proteins.
No, some of it is, most of it isn't.
Okay. How much of the DNA programs for proteins? 4%.
The number I got when I looked into this was 1.15%, not 4%, but okay.
96% does not code for proteins. What's the 96% doing?
I already explained what. Some of it is just to protect the genome, some of it is regulatory, some of it is literally doing nothing but existing.
Answer! It's vestigial. It's doing nothing. It was called junk DNA.
I guess I can think of some of it as vestigial, although that term usually applies to organs, not genes. But also no one thinks that the dichotomy is protein coding versus literally nothing.
That was its official title in evolutionary biology. Junk DNA. What a spectacular example of God not doing it. 96% is junk, only 4%. But evolution predicts this.
Well, it predicts that some DNA will be useless, but not exactly how much in a given organism.
That DNA comes from some primitive animal ancestor, which came from a squid, which came from a clam, which came from a colony of bacteria.
There are no squids or clams in the ancestry of non-mollusks.
The DNA is pulled along, tweaked here and tweaked there to improve itself, and all the old stuff is just carried along, to the point where 96% of it is just dead weight, doing nothing. About 50 years ago, this started to come apart because people started to find functions for the so-called junk DNA. A paper came out in Nature, this is only about 3 years ago, a paper in Nature with 350 authors.
Put in the comments your prediction as to whether Rabbi Dr. David Gottlieb will actually give the name of the paper.
Science papers sometimes have a lot of authors, but this is a worldwide effort. Charting chemical activity of the DNA. So far, they have found that 80% of the DNA is chemically active.
Ah, so I do know what paper this is. It's the preliminary report from the ENCODE project from 2012, titled An Integrated Encyclopedia of DNA Elements in the Human Genome, and the listed authors are basically just everyone who worked at ENCODE. Now, ENCODE's mission is to make a full map of the entire human genome, including what every bit of it does, whether that's useful or not. Also, I should point out that the 2012 paper claimed about 80.4% of the DNA displayed chemical activity, meaning that 19.6 did not. So even that report wasn't saying there was no non-functional DNA under their overly broad definition. And of course, only a couple years later, they walked back their functionality claim considerably, and now it's in line with previous estimates of functionality based on things like knockout tests in rodents, where big sections of the genome are simply removed, and the mice are functionally indistinguishable from other mice with intact genomes, and in fact go on to reproduce with other mice normally.
If it's chemically active, then the assumption is that it makes a difference in what goes on in the cell, in which case evolution can apply to it, selection can apply to it, and then the idea that it's functionless becomes very, very hard to swallow.
That isn't entirely wrong. Much of the chemical activity in a cell is spurious, simply because chemistry is going to chemistry, whether it's for the good of the cell or not. This means that things like spurious transcriptions will always occur, no matter how much the cell does or doesn't need it. This means random bits of the genome will be transcribed to RNA that then just doesn't do anything and eventually breaks down. The 2012 ENCODE paper would call that function. Similarly, if part of the genome is methylated, that makes that part of the genome nearly impossible to turn into RNA, rendering it quite functionless. And yet methylation is biochemical activity, and so the 2012 ENCODE paper counted it as function. Basically, the definition of function used by ENCODE was that if X does Y, then Y is the function of X. So to bring it outside the realm of DNA, if a hot pan burns your hand, then the function of hot pans is to burn your hands. Of course, we know that that's not true. That's not why humans heat up pans. They do it to prepare food. That's the function. The burning of hands is an unwanted and often avoided side effect. Or there might be something with no practical use. Perhaps your grandmother left you her porcelain dolls, and you just kind of forgot about them in a display cabinet in the guest room. Now they collect dust. So the function of your grandmother's porcelain dolls is to collect dust. Well, no, they don't really have a function, and the fact that they're collecting dust is actually evidence of that, just like methylation in DNA is evidence of lack of function. And of course, just remember that David here isn't mentioning that ENCODE corrected this paper only a couple years later, and that the findings made a splash not because of how amazing they were, but because of how obviously wrong they were. Something ENCODE now agrees with.
But they have found specific cases of non-coding DNA, and they know what the function is.
Yes, one of the biggest functions of non-coding DNA is to serve as a promoter for coding DNA so that the DNA is expressed. This isn't news and doesn't negate the fact that the majority of the human genome doesn't just not have a known function, but instead is known to be functionless.
Here's the one that I find spectacular. 96% doesn't code. Take the 4% that does. Little stretches, which the RNA copies and then makes proteins out of. But guess what? In the stretch that does code, there are little pieces that don't code either, called interons.
Well, it's intron, not interon, but yes, this is true, and it's characteristic of eukaryotic genomes. Prokaryotes don't have introns. Now, the ends of introns on either side are functional in that they inform the machinery of the cell to cut out the intron. But in the stretch between these cut here markers, the DNA is just a mess of random mutations and transposons, which are basically useless genetic parasites, also repeats, etc. They are very clearly not functional stretches of DNA, and the fact that they are transcribed only to be cut out indicates this rather well.
They're just junk in the way. So here's what happens. The RNA copies it, including the little pieces that don't code. Then the RNA splits up into pieces. The little pieces that don't code drop out. The other pieces sew back together, and that sewed back together goes over to the ribosome and creates the protein.
I'm impressed. Compared to the rest of this, I can tell that David has actually bothered to learn about how eukaryotic transcription and protein synthesis works, at least in some detail. If only he could have this level of familiarity with the rest of what he's saying.
What a kludge! What a Rube Goldberg procedure! You got the thing that codes with things that don't code, copy it all, detach, fold out the ones that don't code, then reattach it together. What a stupid, inefficient system.
In fact, yes, it is pretty inefficient, and we can tell because the organisms that don't do this, such as bacteria, are measurably more efficient when it comes to protein synthesis. There's a reason that when an organism was chosen to be genetically engineered to produce human insulin for use in type 1 diabetics, it was a bacterium and not an animal, plant, or fungus.
Guess what they discovered? Yeah, the little pieces that drop out don't code. But the rest of the pieces that do code, that you sew together to make the instructions for making the protein, you can sew them together in more than one way.
Which still requires more energy than just having separate genes for the separate proteins that lack introns, and still doesn't make anything but the end bits of the introns functional. Introns could be much smaller and lack things like transposons and stretches of noise and still perform this function, which again is still less efficient than it could be. That introns as such have some function doesn't mean that all DNA in an intron is functional.
You can sew them together in 20 different ways and make 20 different proteins. That means the single section of the DNA is coding for 20 proteins. Not one, but 20. That multiplies the informational content of the DNA by 20.
Oh hey, an anti-evolutionist with a definition of information. I guess information is how many proteins you can make with a particular stretch of DNA. Kind of a weird definition, as I'd expect that proteins of different sizes to be different amounts of information, but I guess not. Let's just hope he sticks with this definition of information.
The functional information content. And guess what? The little pieces that drop out sew the other ones back together.
Kind of, but again, the middle bit of introns is just noise. So at best you can say that there is function at the head and tail of the intron, but not all of it.
They determine the orders in which the other ones can be sewed together. Are they useless? Are they junk? No, they are not. They actually multiply the coding capacity of the DNA by 20.
Yes, as a whole, introns are not junk, but they contain junk because most of a given intron is going to be functionless, because the length and sequence of introns is essentially unconstrained, except for the small functional bits at the end.
So they don't code. So what? That doesn't make them useless, functionless, junk.
Correct, non-coding is not synonymous with non-functional, but neither does that mean that all non-coding DNA has function. One of the ways we can tell if DNA has function is whether it is under selection pressure. If DNA is functionless, then it doesn't really matter what the sequence of that DNA is, or exactly how long that DNA is. Whereas if it has function, then those things do matter, and we can check the genomes of organisms, humans included, to see which parts of the DNA are so constrained, because such regions of the genome will be highly conserved, not just within species, but even between them. We also observe that most of the human genome shows no evidence of being conserved by selection, meaning that it doesn't matter to the functioning of humans what's in that DNA, meaning that DNA is functionless, since it can be literally anything or nothing with no effect on the organism. That's why I can say with confidence that much of the human genome is said to be functionless, not simply because we don't know of a function for it, but because we know it not to have a function. This isn't just default to no function until we can show otherwise. This is we can show no function positively.
Another thing, these complicated molecules, like proteins, and certainly like DNA, you've seen the beautiful graphic accounts of the double helix of the DNA. But of course, DNA never exists as a super long chain, yards long, of a double helix. It's all bundled up together, wound up on itself over and over again.
I think he just means that DNA exists in chromosomes, which for eukaryotes is true.
Proteins are that way too. They're all wound up on themselves. How do they wind up?
Chemistry. Different amino acids have different electrochemical properties, meaning that they attract or repel other amino acids in a protein, causing the whole thing to fold in on itself. Now, sometimes shepherd proteins are used to reduce the energy needed for these folds, and the folds are so complex that we do not currently have a working mathematical model that can successfully predict how a protein will fold just from the amino acid sequence. But we do know that the answer is chemistry, not magic.
What makes them wind up? Does it matter how they wind up?
Yes, the way a protein folds exposes various functional sections of the protein to the intra- or extracellular environment so that it can perform its function. Misfolded proteins have functional sites that are inactivated and may form spurious functional sites that may even be harmful, turning the protein into a toxin.
It matters a lot. If it winds up the right way, then it does all the things it has to do. If it winds up another way, it won't do those things.
It might even do harmful things. That's why protein coding regions tend to be highly constrained and show lots of evidence of selection, as do associated non-coding regions such as promoters.
Sometimes it'll do nothing, and sometimes it'll do destruction. It's got to wind up the right way. What winds it up? There are no spools in there winding it up. No. The molecules that make up the DNA have magnetic forces.
No. In order for the DNA, which doesn't have any significant magnetic force, to be the cause of protein folding, the DNA would have to be present where the folding occurs. But in eukaryotes, the folding occurs outside the ribosome, which itself is outside the nucleus, which is where the DNA is. The DNA isn't there to fold the proteins. I can't believe I just said that this guy seemed to have looked into this and have a pretty decent understanding, only for him to then go on and say that proteins fold because of magnetic DNA.
And they stick to one another in a particular way. It's an unsolved problem. How something that's thousands of links long folds up in a fraction of a second the right way every time. No one knows how that happens.
Actually, it doesn't happen every time, and we do know how it happens. It's chemistry. Positive charges and negative charges attract, and boom. It happens for the same reason that water molecules attract each other's positive end to negative end. The mechanism is well understood. What's lacking is a way to predict in the highly complex environment of a protein chain exactly what folds will occur. That's the unsolved problem. No one is wondering what forces are at play. It's similar to the three-body problem in physics. Everyone knows how orbits are established. Gravity. But in gravitational systems with more than two bodies, the interplay of forces is extremely complex, and so there is no single equation that can simulate n-body gravitational systems continuously. Instead, the only way we've found to simulate gravity with more than two bodies is to simply sum the forces at a certain point, then move the objects in the simulation a distance by skipping forward in time, then resumming the forces. This is accurate enough if the time steps are short, but it's never completely accurate. But that doesn't mean that maybe orbits are actually not the result of gravity. Similarly, we know how proteins fold. We just can't predict it exactly.
But that's how it happens. But what holds them together is the magnetic properties of the molecules. So a lot of the molecules are in there to make sure that it folds the right way.
Well, electromagnetic, and the molecules are the ones in the protein, not the ones in the DNA. But then, yes. And the thing is that this is selectable, because as David has already pointed out, improper folding in a protein means that it will no longer function as it did originally, meaning misfolded proteins can be selected against. No one has to understand protein folding for natural selection to work on it. That's one of the great things about natural selection. It works whether it's understood or not. You might say that it works in mysterious ways.
They don't code for protein, but so what?
What's the they here? The proteins? They do not code for protein. Or are we back to thinking that DNA has magnetic forces that fold proteins, which again, it categorically does not?
It's got to fold the right way, or otherwise it won't do anything. Any molecule that's there to make sure it folds up the right way is functional. It's not junk.
Yes, and literally none of these molecules exist in the DNA because all of the molecules that assist in protein folding are themselves proteins that exist outside of the nucleus.
And so it goes. Every week there's another discovery as to what the DNA, what the pieces of the DNA are doing. So from the idea that 96% is junk, now that idea is on the run. Most have said we shouldn't talk about junk DNA anymore. It's just a mistake. The whole thing's a mistake, and it's an open field. What the DNA does.
No, that's completely wrong, and even ENCODE says it's wrong. And further, I'm not sure where he got the idea about 4% of the genome is coding. It's less than 2%.
Now I'm going to tell you a footnote. Many people have heard of this. It was very, very widely publicized that human beings and chimpanzees share 98.6% of their DNA.
Are we about to get a Tompkins reference here? Gee, I hope not. I picked this guy in an effort to see what non-Christian anti-evolutionism was up to. Apparently the answer is parasitizing Christian anti-evolution, so that's great.
So that was cause célèbre.
He should work on his French.
And I will take just a moment to explain the philosophical problem here. Look, the difference between us and chimpanzees is 1.4% of the DNA.
In alignable regions, which is important because they are alignable primarily because they are constrained, which means these are the regions that are likely functional. There's no simple way to just get a single number for the whole genomes of chimps and humans, in part because the genomes aren't even the same size. That's why DNA comparisons always come with an explanation of what's actually being compared. At least they do in the actual literature. What filters down to the news outlets and then to people like Gottlieb often loses that nuance. Maybe the nuance is unwelcome.
Now listen, that's science. That's a number. 1.6 is precise. Seems to me that human beings and chimpanzees are very, very different. But I have to be wrong. That's just an impression.
They are very different in some ways and very similar in others. Biochemically, they're almost identical, to the point that vets treating captive chimps can use human blood for transfusions. DNA similarity on its own won't tell you much about the morphological or behavioral similarities between two organisms. Although it'll tell you something about it. It turns out that relatively small mutations in certain developmental or regulatory regions of the DNA can have significant morphological, cognitive, and behavioral effects on an organism.
That's just a feeling. 1.6 is a number. It's a precise number. So that has to be right. So my feeling that we're so different is wrong. We're really very, very much the same. 98.6% the same. Because the scientific picture is a number. That number is precise. Well, first of all, the conclusion is stupid.
Oh, it's stupid. Are scientists meant to be poopy heads? Do we need a timeout to calm down? Maybe some milk in a box? But okay, that's enough old man yells at science for me for one day. When we come back, we'll look more at the chimp-human similarity and difference, and how badly incorrectly David can characterize it. If you've enjoyed the video, hit like and tell me in the comments. If you didn't, feel free to hit dislike and tell me what the problem was in the comments. Either way, please remember to subscribe and hit the bell icon so you're always notified when I have more content. I'm the Dapper Dinosaur. Hey, before you leave, I just want to take a second to thank my patrons and channel members, especially those pledging $20 or above. Benthoven, Tapioca Weasel, Whispers, Kelvin Brostick Van Manen, Denny5252, Eleron Teller, Phil Rivara, Ian Chen, Landon Knoll, Mabbity Babby, Monkey They Them, Mrs. Spexinder, The Venerable Bede, Sphincter of Doom, and Star Runner. It's because of my channel members and patrons whom you're seeing on screen that this channel can stay afloat. Without you, it would all shut down. If you want to join the team, there's a link to join the channel below this video, and there's a link to join the Patreon in the description. On the Patreon, you can get a 10% discount for pledging annually, and either way, you get early access to virtually all of my scripted videos, often 3 to 5 months before they come out for the general public. Thanks for watching.